Hello everyone of the Atlantic community. My name is Júlia Petroski. I am an oceanographer and master student at the University of São Paulo, Brazil, working at the Fisheries Ecosystems Laboratory, whose supervisor is Gasalla. The work that I developed is called Trophic Row of pelagic fishes from the southeastern continental shelf of Brazil, integrating data from historical cruise. I studied the diet composition in fisheries of pelagic fishes in southeastern and south coast of Brazil. Most species in the region are small pelagic, such as sardines, which are short-lived, strategistes, and responsible for a large part of the fish production. So most of the species studied are important economically in the area. It is important to know the feeding habits of organisms to understand trophic relationships, that is, who is the prey and who is the predator in the ecosystem. With this, we are also able to understand a little bit the effects on the ecosystem of overfishing certain species. Knowing the trophic role of pelagic fishes is very important when you think about the ecological studies, since each species causes a different effect on the food chain, but it can also help in fisheries studies in the context of sustainable fisheries. The area studying this project is known as the Southeastern Continental Shelf of Brazil, between Cabo Frio and Cabo de Santa Marta Grande. The ichthyofauna was collected aboard the South Atlantic in the COSAR 2 program, which was a cruise resulting from a partnership between FURG, IAUSP and IBAMA, and took place between June and July 1995. More than 400 stomachs of 12 species were analyzed, which included the Brazilian sardinella, large-head hairtail, Atlantic moonfish, and others. To analyze food composition, diet similarity was calculated for all species, the frequency of occurrence, and for some species was able to use other indexes such as numerical frequency, visual volumetric, or gravimetric. The trophic levels represent the position the food web that each species occupies, ranging from 1 to 5, and were calculated using the trophic level software and the food indexes. 9 of 12 fish species were considered planktivorous, feeding on phytoplankton or zooplankton, and 3 are piscivorous. Through the results of diet similarity, the species were grouped into 3 ecological guilds: planktivorous carangidae, planktivorous clupeiforms, and piscivorous. For the clupeiforms, zooplanktonic organisms stood out in the diet, and among these, copepods and marine shrimp. For the carangidae species, copepods were highly frequent, as well as the small crustaceans. Now, for the guild of piscivorous fish, we saw a high intake of teleoster fishes. As for trophic levels, these ranged from 2.98 to 4.5. These values agree with the guilds and show that these fish occupy an intermediate trophic position, being omnivorous and piscivorous respectively. To investigate fish stocks and their fluctuations, latest data from 1986 to 2019 was gathered through literature review and databases covering the states of São Paulo, Rio de Janeiro, Paraná, Santa Catarina, and Rio Grande do Sul. Comparing the annual length averages in 2 study periods, we observed that 6 species decreased and 3 increased slightly. The most caught is the Brazilian sardinella, and its length has decreased by 38% of these years. Verifying the correlation between the lengths of Brazilian sardinella with other species, there is the possibility of fishing for small pelagic being more intense in the periods of shortage of sardine. So when the sardine abundance decreases, the fleet tends to the capture of alternative species. This study described the feeding habits of small pelagic on the Brazilian coast, in addition to complementary fisheries data, which can fill important gaps for bioeconomic modeling in the area. The scarcity of information on the food ecology of the most of the species studied here points to the need for more studies that also cover spatial and temporal variations and a joint analysis to investigate the fishing capacity with changes in the structure of the food chain. Thank you so much for your attention. I would like to thank the National Council for Scientific and Technological Development, CNPq, for the financial support to this project and to all those who are involved in this work.